Okay, so before we get started today, as I've said before, I do like to put up services for students. So one of the services that can be quite useful in this stressful time of self-isolation is the ASU counseling services. I do know that they're doing this also remotely. And this is at minimal charge to students. In fact, I think for most students it's free, or there's a $10 per semester charge for counseling services. So if you're feeling stressed out, please take advantage of that. Okay, so let's start with the bonus question from last time, which was, if an unnamed professor accidentally stuck themselves in the thigh with a syringe containing a virus, which lymph node would you expect to dramatically increase in size? And that would be indicative of a T cell and B cell response and generation of adaptive immunity. But the second part of that question is, 10 years later, would you expect to find memory T cells specific for that virus in the spleen? Okay, so for the first part of it, where would the response occur? Well, if it's in the thigh, it would be traveling towards the heart, and the first lymph node that it would encounter would be the inguinal lymph nodes. These are, in humans, sort of on the inside part of your hip. And in fact, what happened is, I can tell you from personal experience, is that that lymph node swelled up to about the size of a golf ball. So it was a very strong immune response. And so that indicated there was a massive T cell and B cell proliferation in that lymph node. Now, the response can continue upstream. If the virus continues to spread by the lymphatics, you would get involvement with other lymph nodes on the way up to the heart. It would not occur in the inguinal lymph node in the other leg, because that's not upstream. It's still downstream, or it's at the same part of the stream, but on the other side of the body. It also wouldn't occur in the popliteal lymph nodes, which are behind the knee, because that's not going towards the heart, it's going away towards the extremities. Now, the other part of this question is, would you find later memory T cells in the spleen? Well, remember, what do memory T cells do after you've made them? They continue to circulate just like any other T cell looking for pathogen. And so they would be moving from the blood into lymph nodes, eventually traveling through the lymph nodes, going to the...
Hey, what the?
Everybody okay?
Yeah, I'm so sorry. That was my cat. He just knocked something off the shelf.
It's okay.
I thought I was on mute.
It's okay. So memory T cells circulate just like any other T cell. And so some of them would be going through lymph nodes and some would be going through the spleen. So yes, you would definitely see them in the spleen. You'd also be able to find them in the blood. And in fact, Alexander Seti at Scripps Research Institute did a study of people that were infected with this virus and how much of the immune response was specific for it. And in my case, I have about 4% of all of my CD4 cells are specific for this virus, and about 5% of my CD8 T cells. Interestingly, after you wrote the paper, the journal sent it to me to review, which I had to recuse because I'm one of the patients in the paper. Okay, are there any questions about that bonus question? All right. So today we're going to begin to start talking about how the innate system recognizes foreign pathogens and how it sort of tailors the response to give you the kind of response that would be most beneficial to that infection. And this will be a recurring theme throughout the semesters. How does the immune system know it should make a response? We recall the innate system has two jobs. It provides the first initial response to infection to control it as best it can. And it also tells the adaptive system, hey, there's something here, you should make a response to it. But the problem is that we don't always want responses. If you consider yourself as an organism, you actually have more bacteria on you, or bacterial cells on you, than you have of your own cells. So these are the concentration of bacteria at different sites in your body. On the skin, you have about 100 million bacteria per inch. In the small intestine or the large intestine is quite higher. You can have up to a million per milliliter. So there's quite a lot of bacteria, and we don't always want to make responses to that. And if you consider, you have about 10 to the 13th cells in your body, but you have about 10 to the 15th bacterial cells, you're only really 1% human in terms of cell number. And you also have lots of viruses, you have fungi, many of you might have parasitic infections, and so there's really a lot of other stuff that you don't necessarily need to make a response to. And in fact, many of those, particularly bacteria or lactobacilli in the intestine, are very beneficial to you. They help your digestion, and if you don't have them, you actually develop pretty severe inflammatory bowel disease. So the question is, how does the immune system know when it should make a response? And how does the innate system tell the adaptive system, this is the type of response you should make? And then ultimately, how does it minimize collateral damage? Because the immune system is not just a benevolent force that goes around and kills pathogens. It's trying to make you inhospitable to whatever's infecting you. And so you don't want to kill yourself in this process, or you don't want your immune system to kill you. Okay, so if we go back to sort of the problems that the immune system is facing, is that microbes reproduce much faster than we as humans do. We have something like, we have a 9-month gestational period. And if you consider maturity to breeding age, then we're actually looking at decades. Viruses and bacteria can do this in hours, sometimes days, but it's still much faster. And because of that, the microbes can evolve or adapt to the environment much faster than we can. So the immune system has to have a way of doing this in the same sort of speed. And we'll talk about that as we go through adaptive immune responses. The other problem is that you only really have about 20,000 genes. And if you consider the millions of different potential pathogens, you can't just have a dedicated system for each pathogen. You have to have sort of a way of evolutionarily responding to anything that comes in. Okay, not only that, but if you dedicated one gene per microbe, even if you were able to cover everything, microbes would evolve and change it, and so you wouldn't be able to keep up, just because you can't replicate as fast as them. And so what happens is that your innate system recognizes general immune features or general pathogen features that, in general, it's very difficult for that microbe to completely get away from it and still be a viable organism. It's recognizing things that are very common to many different families or orders of microbes. And these are things typically that you don't have on your cells. So the 3 classes of things that you're going to recognize are lipids, sugars, or nucleic acids. And it's different for different types of infection. For bacterial infections, typically you're going to recognize lipids and sugars. And in particularly conserved lipids and sugars that are present on many different types of bacteria. For viruses, because viruses use your own cells and your own membranes, they look a lot like you. So it's difficult to use the sugars and lipids. Instead, what the innate system uses is the presence of nucleic acids that you don't have or that are in the wrong place in the cell. Now, the innate immune system is much older than the adaptive immune system, evolutionarily, right? So all animals and plants, even some microbes, have an innate system. So if we were to go out and look at a tree, it would have some version of innate immunity. It produces things that inhibit things that want to infect it. We now know that that some bacteria have an innate system where they'll take in little bits of bacteriophage genomes and make an intracellular response that will limit bacteriophage replication in that microbe. So innate systems are quite old. Now for plants and most non-vertebrates, the innate immune system provides the most of the burden of protection. And if it fails, then the host dies from infection. The main reason for that is that an adaptive system requires a circulatory system so that you can constantly move around these cells and survey if there's an infection. The innate system is a little different. It's mostly tissue resident. So we go back to our tree example, it's going to be just underneath the bark. If something gets past the bark, there's the innate cells and the innate molecules that will provide protection. So question, do sharks have an innate and adaptive immune system? Just give me a thumbs up if you think they do. They have both. And I would say that these little questions, this is one of the benefits of coming to class. This is sort of questions that I might ask on an exam. They don't appear in the PowerPoints that I post on Canvas. This is just what you get for coming to class. Okay, so it looks like a lot of you think they have both. And in fact, they do. Sharks have a circulatory system. So they can maintain an adaptive immune system. So basically, if an organism has a circulatory system, it's very likely that it also has an adaptive immune system in addition to the innate immune system. Now, in vertebrates and some invertebrates, the innate immune system does provide the first line of protection. And we know that that's important because if you are, for example, neutropenic, you're not able to control fungal infections very well. And so that innate system does provide the first line of defense. But the second job is to inform the adaptive immune system to make a response. And it does this by recognizing these conserved structures, and then these innate cells begin to produce pro-inflammatory second messengers that activate B and T cells, or they activate dendritic cells that then all the cells to go to a lymph node and make a response. Now, this is a key step in the generation of an adaptive response because the type of recognition, the type of cells that are responding results in production of different inflammatory cytokines or proteins that then modulates the adaptive response. So you'll get different kinds of cytokines or second messenger proteins when you have a bacterial infection versus a viral infection. And there's quite a lot of overlap, but in general, that's how the innate system is telling the adaptive system, yes, you need to make a response, but also this is the type of response that would be most beneficial. Now, if we go back to sort of our barriers to infection, the key step for any microbe is to cross the epithelial barrier. If it's maintained at a mucosal surface, it typically is not a problem unless something else is happening. And so this is often because of physical damage. So for example, a break in the skin, but also could be loss of mucosal defense mechanisms. For example, if you take antibiotics and you reduce all of the commensals in your digestive system, you become susceptible to some Candida species of fungi. And so it's really any loss of those barriers really can result in access of the microbe to your system. Once that happens, then that microbe is no longer fine, it becomes a pathogen. So this is sort of where the line blurs between a commensal or something that's just sitting around in the gut. If it gets access to your system, then it becomes a pathogenic organism. An example of this would be when you have small bowel resection during some intestinal cancers. And a major problem with that is that then you can have spillover of commensals from inside, from the luminal side of the intestine into the body cavities, which then can produce a pathogenic infection. We often call that opportunistic infections. Now this is a big problem in many other diseases that you don't normally think of. So I think you've probably all heard of cystic fibrosis. This is a eventual scarring of the lungs, which ultimately results in inability to exchange oxygen and then death. And this results from a defective chloride channel. And you wouldn't think that's a big problem, but what happens is that you are unable to move the mucus from the lungs into the oral tract. We often call that the oral toilet. So if you're coughing up phlegm, it then goes down your digestive tract and you destroy it. And so because it can't do that, then you get growth of bacteria in the lungs, and you're constantly making immune responses to that, and that eventually leads to immune damage of the lungs and scarring. So what happens when we think about the innate system? In all of your tissues, you have innate immune cells present just below the epithelia. So anytime something crosses it, you have cells there that can sense it. And this includes granulocytes, in particular mast cells or other granulocytes that have become tissue resident. It includes macrophages that are set up there. It includes dendritic cells. There are also other populations of NK cells and gamma delta T cells that are set up there ready to respond. And if they sense something, then they alert other cells to come here, check it out. And they do this by secreting the chemokines that we talked briefly about, and other cells undergoing diapedesis to that site. Okay, so let's talk about sort of this opportunistic infection. The first thing is you have to be able to get to the skin or to the epithelium. And in this case, we're talking about the epithelia of the skin, and you get a cut. And assuming you didn't have somebody wash that out with hydrogen peroxide or some other sterilizing agent, that infection is going to gain access past the dead epithelial layers of the skin or the epidermis and get into the system. Now, once there, it can be sensed. Okay, so it's going to be sensed by bacteria or by macrophages that have all of these different receptors on it. It's going to be sensed by the granulocytes that have receptors for all these things. And it's also going to be taken up by dendritic cells. So what happens then? Well, these cells that have sensed the infection, even if the wound is repaired, there's infection underneath the skin, they're going to alert other innate immune cells to come here. And so they're basically saying, cells from the blood, get in here. Okay, so that would include more of sort of the effector arms of the innate system, including the granulocytes in particular, neutrophils, etc., which will go there and sort of try to eliminate the pathogen on their own. Meanwhile, dendritic cells that sense the infection are going the opposite way. Right? They're going from the skin to a lymph node to tell the adaptive immune system, hey, we think there's something here, see if you want to make a response to this. Once that happens, you have a response in a lymph node. So it goes down here to a lymph node. And you make a B cell and a T cell response. And the B cells will make antibodies that can limit the spread of infection. But in particular, the T cells will return to that site and begin killing off the infection. And this is true for both viruses and bacteria. So you sort of have these 4 different steps, okay? You have one, if it's outside of the epithelia, you don't sense it, or the immune system doesn't sense it. Once it gains entry because of damage to the epithelia, it's going to start replicating. And the more it replicates, the more signal there is for the innate system to make a response. But ultimately, clearance of the microbe really depends on this adaptive immune system that is generated in a lymph node. And so you sort of have, the blue is what the microbe is doing, but the red is sort of what the immune system is doing. So once there's infection, the innate response is sensing that. The dendritic cells and other cells are signaling to the adaptive system to make a response, and the adaptive system comes in and provides an overwhelming response that tries to eliminate the pathogen.
I had a quick question.
Sure.
You used the term effector function of the innate immune system. Is that just granulocytes or?
Well, granulocytes can do it, but remember, macrophages can phagocytose stuff. Although we think of them as the garbage trucks of the immune system that come in and clean up, they're also sensing things and making cytokines, which are effector mechanisms. So the granulocytes are sort of they're degranulating. They're sort of trying to isolate the infection. Particularly the neutrophils are making these nets. So we think of granulocytes as more of the effector arm of the innate system, but also NK cells would come in and kill any infected cell. Or start to kill the infected cells. Okay, so one of the terms that we're going to use a lot in this course is a PAMP. And this is a pathogen-associated molecular pattern. They're not rigidly defined. So you have the microbial structures typically will have certain variations on these. And your innate system has evolved to sort of recognize a general structure. Now, an example of this is a protein called a toll-like receptor, or toll-like receptor 4, which recognizes different versions of lipopolysaccharides. It recognizes tetraacylated, pentaacylated, and triacylated forms. And so it's just a general structure that it recognizes. And remember, these are things that a microbe can't change very easily, right? It needs that for other parts of its biology. And so if it was to change that to something completely different, it would essentially become a different organism. And that's not easy to do. And so what you have are PAMP receptors, or pattern recognition receptors. These are the proteins on your innate cells that recognize foreign things. And we talked about some of them, like scavenger receptors, et cetera, but we're going to go over them again. Okay, and so the idea is if you have some innate cell, let's say it's a granulocyte, they're going to express these pattern recognition receptors that are going to bind to these conserved structures on the pathogens. And it results in a stereotypical response. So typically, if it's recognizing sugar or a fat, or lipid, should say, it's going to assume that that's an extracellular pathogen. If it's recognizing a nucleic acid, typically is going to say this should be an antiviral response. Okay, so just that first step of what is it recognizing tells the innate cells what kinds of cytokines they should make in order to direct the response to be most beneficial. Okay, so for example, here we have a viral infection. The virus gets into the cytoplasm and you have viral RNA. And often this is double-stranded RNA, and that's going to be recognized by proteins like RIG-I, which we'll get into a bit later. Now, why does that work? Because you don't have double-stranded RNA in your cytoplasm. And so that's going to set the cell off saying, there's something foreign here. On the other hand, if it's recognizing bacterial lipopolysaccharides from extracellular receptors, that then gives a different signal to give production of different things. So in general, nucleic acids will give you things like interferon, type 1 interferons. Extracellular things will give you things like IL-6 or IL-12. Those different cytokines then direct the immune system to make different kinds of responses. Now, there's two different layers here. One is that the pattern recognition receptors, right, that recognize PAMPs, are saying this is something foreign. So that sort of plays into self versus non-self discrimination. However, it also tailors the response to give you the best type of response. So if you don't recognize anything, you don't make a response to yourself that maintains tolerance. And then you can have different types of pattern recognition receptor signals that then provide different signals to the adaptive cells. Now, when we talk about autoimmunity later in the semester, we'll see that this breaks down quite often. And in fact, if you have a lot of damaged tissue and you have cells that are dumping out DNA into the extracellular environment, that can be sensed too. And that's a different version of that. That's called damage-associated molecular patterns. And that's really where self-tolerance breaks down. Okay, so these conserved structures, as I said before, they're very common in many microorganisms. And if you mutate them, then you get significant replicative cost. You can think about this: if it's a virus, and it would have to change what its nucleic acids are. And it's very difficult to do that because you have to essentially become a new organism. For yeast and bacteria and parasites and other microorganisms, you've got these, they've got a different structure, right? If you have a gram-positive bacteria, then it has all of these lipopolysaccharides on the surface, and that it uses to sort of maintain its own biology. If it's a gram-negative bacteria, then you have lipopolysaccharides on the outside, and then other things on the inside that it needs, and it can't just change those without significant damage to its replicative ability. Okay, so the immune system, the innate immune system, is relying on the microorganism. Definitely needs that. Now, if you have, if you've ever seen the movie The Andromeda Strain, which is a silicone-based life form that comes from outer space, would you have the ability to recognize it? No, because you haven't. It's not anything. It's not related in any way to the microorganisms you would normally encounter, and so you don't have a mechanism for recognizing something like that. Okay, so now we're going to, in this lecture and next, we're going to talk about the different families of pattern recognition receptors. And the first group are the scavenger receptors. And this is really important for both gram-negative and gram-positive bacteria because they recognize extracellular charges or polysaccharides that have these strong negative charges on them. Some of them are recognizing lipoproteins like lipopolysaccharides. And so these are structures that are very common. And I should say they're also not very well defined. You can it's because it's just based on charge primarily. There's lots of different versions that can be recognized. And as we'll talk about when we get to toll-like receptors, some of them are pretty promiscuous and recognize things that you wouldn't think look the same, but to that protein it does. The second group are the lectins. And so the first one are sort of general charge. They're just looking for things that look very different than you. Lectins are recognizing different polysaccharides. As you remember from probably your cell biology courses, right? You attach polysaccharides to your proteins as they go out and are put on your on the cell surface. But they can also, these lectins are really recognizing different polysaccharide structures that you don't have. And this would be particularly important for fungal recognition, but you also use lectins for other things, including just this rolling and sticking that we're talking about, the cell rolling along the epithelia. Lectins are a very broad group. They recognize both, they can recognize either self things which results in tolerance, or if they recognize foreign things, they can result in activation of the immune system. And then we get to some that are more specific. And so these recognize much more narrow versions of things. And the first is the toll-like receptor or family. And these are a group of proteins that recognize, some of them recognize extracellular things, But there are also toll-like receptors that are kept in endosomal compartments. And so if things are brought in and they start to get digested, then they're recognizing those in the endosomes. Inside the cell, you have RIG-I-like receptors. And some people will further break these up into DNA receptors and RNA receptors, but I'm just going to keep them as one group. And it's named after the prototype, which was RIG-I. This is one of those weird stories that it was actually a font issue. It was initially called RIG-1. Because of Arial font, it became RIG-I. And then finally, you have NOD-like receptors. And these are a little different in that they're really recognizing cellular imbalances. So they're recognizing concentration of ATP. They're recognizing heat, stress, electrical charge, ion differences, release of cytoplasmic calcium, or other things that just make the cell seem abnormal. And we'll go through each of these groups in a bit. But sort of the idea here is that you have a number of different receptors on your granulocytes and macrophages in particular, as well as on dendritic cells, that are able to sense all the different kinds of life forms that are potentially infecting you. If it's a bacteria, you can sense that outside the cell. If it gets into the cytoplasm, you can also detect it. If it's a virus, you can detect it either in the endosomes or inside the cell. If it's a lectin or scavenger receptor, you can detect fungi and parasites that way. And if it's just gets in and evades everything else, still have the NOD-like receptors that are basically there to say, okay, something's wrong, we're just going to make a response anyways. Now the scavenger receptors are pretty diverse. There's literally hundreds of these, and they're really recognizing things based on charge. So it has to be strongly anionic, and particularly polysaccharides, lipoproteins, many of which have very charged heads on them. And they have lots of members, right? I showed this example before. Scavenger receptors, there's several families. Actually, it goes beyond G. I think there's up to group I now. And they have lots of different structures because their job is to recognize basically anything that's out there. And so it's going to recognize sugars or lipids that you don't have on your cells. Now, these can be expressed in a variety of places. And the rationale for this really is that, well, if it was just on the cell surface, what would happen if it escaped into an endosome? And so you have, these can be on the cell surface for many of them, but they can also be expressed in the endosome. So if you start taking something in, that granulocyte or macrophage knows, hey, there's something else here, potentially going to be infecting this cell. And it's really just coverage. You want to be able to recognize these wherever they may occur. But the scavenger receptors, if you think about the name, let's go back to our macrophage example. The macrophages are the garbage trucks, right? And so if you don't have these, some of them are specific for yourself or bits of yourself. And so they can recognize sometimes both the host and the microbe. So an example down here would be CD36. It can recognize PAMPs on bacteria, but it also can recognize things from your dead or dying cells. And what that does is if it's recognizing your proteins, it actually provides a tolerogenic signal. So it's saying, no, don't make a response. But if it's a microbial signal, then it says, yes, you should make a response. Now, the problem with that really is it's all in context. Is there another inflammatory signal? Is there another cell that is in that environment that's saying, yes, this should be a response? And if there's not, then you don't make a response. So scavenger receptors on their own don't really set off the innate system to say there should be something here. They're mostly just for cleaning up stuff. And so if you have sort of the scavenger function, right? The macrophage is sitting here and it binds to self-protein, well, that dead cell is actually providing, in turn, a signal to say, no, I'm just a dead self cell here that you don't need to make a response to. Now, when you're studying for this, I don't care that you know all of the scavenger receptor names, what the other signals are. What I care that you know is that without an inflammatory response, without inflammatory cytokines, and the provision of these, yes, I'm just a self cell, don't get too upset, that maintains tolerance. But if you look on the left, if it instead binds to something that's foreign, in this case Staph aureus, then it can bind that, but you get other signals, and typically it could be on the same cell or other cells, and that results in the production of pro-inflammatory cytokines. In this case, IL-12 and TNF. And those are strong signals that tell the cells, yes, you should make a response to this, get everybody engaged, and let's get going. Okay, so scavenger proteins have sort of this dual function, and it really depends on the context in which they recognize those things that tells the macrophage or granulocytes what to do. Okay, now that's really all that I care that you know about scavenger receptors. Their job is both in scavenging dead cells and sort of wound healing processes, but also recognition and internalization of foreign things. And in that case, it's the context in which they see the microbes. Lectins are very similar. So they recognize, as the name implies, polysaccharides. I don't know if any of your parents ever did canning, but you add lectin to, when you're canning, to preserve sugars. And so it's really lectins are sugars, or lectins recognize sugars, and it can be on free proteins. Okay, so if you're making proteins and they have lots of sugars on them, your cells can recognize those, and they basically act as a scavenger for extracellular stuff. And again, that doesn't provide an inflammatory signal. It's just saying, here's something we need to clean up. They can also be important for host cell-to-cell signaling. And in particular, they're really important for cells sticking to each other. If you have lectin on one cell, it's going to recognize sugars on another cell, allow those cells to stick together. But if it's recognizing sugars on a foreign thing or a microbe, then it's going to have this role of, okay, there's something here, I need to make an innate response. Lectins separate this more by which protein is being expressed. Okay, so a lectin is specific for yourself or it's specific for something else. It doesn't really have this dual recognition function. Each one, although we group them all together, they each have more of a clear-cut role or a clear-cut job. So the scavenger lectins, not to be too confusing with scavenger proteins, it's really recognizing your self stuff. They take it in the endosomes, they break it down, and they reuse the parts. In the host, sort of cell adhesion, it's really the cells are talking to each other, and these are host cell-to-cell contact. But some of the lectins recognize polysaccharides on bacteria and fungi, and once that happens, this will cause for some of the first steps in immune activation. So if we go back to the scavenger receptors, this provides context. The cell's got scavenger receptors, it's got lectins, and the lectins sort of activate the cell to say, yes, you should be getting angry about your scavenger proteins taking these up. Lectins turn out to be really important for another pathway called complement. And complement is perhaps the most underappreciated part of the immune system, but it's really important for much of what we'll talk about this semester. Lectins are very important for activation of complement. And so some of the lectins that bind to microbes specifically will activate complement to promote inflammation. And one of these is the mannose binding protein, or mannose binding lectin. This is a serum protein. It's soluble in your plasma, and it will bind to foreign things to activate complement. Now, for the lectins that recognizing either foreign or self things, they can recognize these and then result in immune activation. And in particular, what they're doing is their distribution of these is on specific cells. So granulocytes have a different set of lectins than macrophages do. That's because they have different jobs. Okay, so macrophages are more like the garbage trucks, and their job is to come in and clean up the mess. And so they're going to have more lectins that are for self things. Whereas granulocytes don't really clean up the mess. In fact, they cause a lot of the mess. And so what they're doing is expressing lectins that recognize foreign things. In any case, lectins, their main job really is to bind to stuff, bring it in, and have it phagocytose. They then can signal. So if you see a lot of these have signaling domains on them, and they can change how that cell is acting, right? So it can tell that cell, yes, you should make some pro-inflammatory cytokines, change what your adhesion molecules are, upregulate co-stimulatory proteins and major histocompatibility complex proteins. And so that's really the role of lectins. And the scavengers and receptors or sort of can recognize things very broadly, but they don't really on their own activate the immune system. Lectins can activate the immune system, but that's because each lectin is more focused on a specific thing. And so it knows if I recognize this, that is a self thing, but if a different lectin recognizes a foreign thing, it knows that is a foreign thing. Okay, now do I care that you know Dectin-1 or Dectin-2 or any of these DC9? No, I don't care. I just need you to understand that lectins bind to polysaccharides, and their job can be either for scavenger, can be for cell-to-cell binding, or it can be for microbe recognition. The next group that we're going to move on to are the toll-like receptors. And what you'll find is that we're getting more specific here. Toll-like receptors typically bind one thing. They might bind different versions of that thing, but they're really looking for one thing. And they're, with one exception, they're almost always for foreign things. But you might be surprised that, in fact, this is not limited to mammals. And toll-like proteins were discovered by Christian Nussbaum-Volhard in fruit flies. And so what they found is that if you had Drosophila that lacked the toll protein, at that time they only knew about one, that they would have abnormal development at the embryonic stage. And so on the right there, you can see a normal fruit fly, but then on the right, you see the toll deficient fruit fly, it just doesn't develop. Now, how does that play into the immune system? Well, she got the Nobel Prize in 1995 for discovering this, but it was really Jules Hoffman that said, okay, if you take those toll deficient fruit flies and you get them to develop, that they are then all of his colonies would suddenly die out from fungal or bacterial infections. And so they had observed a number of toll genes in these fruit flies, and that some of the mutants were susceptible to bacterial infection, but normal resistance to, or susceptible to fungal infection, but normal resistance to bacteria. Others were susceptible to bacteria, but resistant to fungal. And so basically laid the ground that says, we have toll-like receptors are recognizing foreign things. So then we go to experiments by Bruce Beutler, and who shared the Nobel Prize in 2011 with Jules Hoffman. And he made an observation that seemed at the time very difficult to understand. And so there's two strains of mice. There were wild-type black mice and mice that were a specific strain of mice called C3H. And C3H mice have a naturally occurring mutation in the toll-like receptor 4 gene. And so what he did is if he injected live bacteria into wild-type mice, those mice could make an immune response and they survived. But if he injected bacteria into C3H mice, they actually had an impaired immune response and they would die from the bacterial infection. The problem, or the thing that made it confusing, is if you took LPS from the wild-type mice, or sorry, if you took LPS from the same bacteria and you injected a lethal dose into wild-type mice, those mice would die. But if you injected the same dose of LPS into a C3H mouse, they survive. And so what he assumed from this is that the TLR4 was important for making an adaptive immune response, or for recognizing that there's an infection. The difference here is the bacteria will replicate and eventually kill the host if you don't make a response to it. LPS doesn't replicate. So if it's just ignorant of it, it's not going to respond to this lethal dose of LPS and doesn't have any effect on it. So that was really the aha moment that this is the innate recognition that ultimately leads to an adaptive immune response. So this is what toll-like receptors look like. And in this case, this is a toll-like receptor that's recognizing a nucleic acid. So you have sort of in green and turquoise, the two dimer, or the two parts of the toll-like receptor. And these are either homodimers or heterodimers. They have sort of this characteristic horseshoe shape, or sometimes called a shepherd's crux, where it has the recognition part of it looks sort of like two horseshoes put together. So if you see here, what's happening? This is the horseshoe part of it. This is the other horseshoe part of it. That is the recognition part of it. On the inside of it is the signaling domains. This is where it tells the cell what to do. Now the homodimers, it's just two of the same proteins, but sometimes you have heterodimers, and we'll talk about those specifically. They can also have accessory proteins. So toll-like receptor 4 homodimers have a protein that called MD2 that also binds to it, and that modulates what it recognizes. If it's just TLR4 alone, it recognizes certain types of LPS. If it has MD2 bound to it, it can recognize different forms. So this sort of expands the recognition abilities. Now the recognition domain in all of the toll-like receptors is facing outside the cell. And that includes, if it's in an endosome, it's facing the inside of the endosome. It's not facing towards the cytoplasm. And that's sort of shown here. They're compartmentalized. Okay, so here on the surface, we have some heterodimers. We have TLR2 and 6, or TLR2 and 1. And those are recognizing different lipopeptides. This should actually have 2 of these, right? Because they're always dimers. You should have TLR5 with another TLR5, TLR4 with another TLR4, and those are going to recognize extracellular things. And if you notice, what they're recognizing are things that you don't have. You don't make LPS, you don't make these triacyl lipopeptides, so they're definitely foreign things. And so they recognize that one group, and that tells the cell, yes, there's something here. Now in the endosome, you also have these toll-like receptors. 3, 7, and 9 are the main ones. And they're recognizing nucleic acids. And I think people often get confused about this, but why would you have nucleic acids there? Well, what's happening is as stuff is brought in, particularly viruses, are brought into the endosome, they're being degraded. And so you're releasing some of the internal components of viruses or some bacteria, and that's what's being recognized, sort of degraded parts of it. Okay, so extracellular toll-like receptors, typically you're recognizing intact structures. They're recognizing lipids or polysaccharides that are on the outside. And we'll go through these individually. Intracellular toll-like receptors are expressed in the endosomes, and they're recognizing degraded parts of pathogens. And in particular, mostly what they're recognizing are nucleic acids. Okay. I think we already covered this, but the question is, why are toll-like receptors that recognize viruses localized to endosome? Why wouldn't you have these on the cell surface? Anybody can answer.
Is it because infected cell infected with the virus already has it inside the cytoplasm?
Well, it's mostly recognizing things on the way in. They're endosomes, as opposed to exosomes when the virus on its way out. But if you think about, it's a question of efficiency. So if you have all of these on the surface, remember viruses are tiny compared to cells. And so you really want to have it in a compartment that it's a high concentration of virus. If it's a cell that, if it's on the surface, it's going to recognize viruses not very well. But if it's in an endosome, there's two reasons. One is there's a higher concentration of virus because the virus is getting internalized, but it's also now, okay, it might be a problem here because we've got virus inside our, inside of our membrane. So it's really saying, okay, what's the most efficient way to do this? Oops, hold on. What happened here? Okay, so this is something that I am going to ask you on the test, okay? And the most important thing that each of these recognizes is the first thing listed. So we're gonna start off with the two heterodimers. These are TLR1 with TLR2 and TLR6 with TLR2. Now, these both recognize lipoproteins. TLR1 and 2 recognizes triacyl, and TLR6 and 2 recognizes diacyl. Do you need to know the structure of those? No. But if you think about it, they're recognizing very similar things, and that's because they both have TLR2. The way to remember this is 1 plus 2 is 3, that's triacyl, and 6 plus 2 is 8. It's not going to help you there, but they're recognizing lipoproteins. Now, they both can recognize other stuff, and they do this, but less well. What I really care that you know is that they're recognizing lipoproteins. That's their main job. Then if we just go in order, TLR3 recognizes double-stranded RNA. So given that you know that, where is it expressed? Is it an endosome or on the cell surface?
It'll be endosomal.
Right, that'll be in the endosomes. Where are the first two at? On the endosomes or the cell surface?
Cell surface.
Right. Okay, now we go back to TLR4, right? That with or without MD2, it is recognizing lipopolysaccharides. Now, these are sort of a different, this is closely related to the lipoproteins, but they're different structures. And this is basically anything that's more than triacyl lipopolysaccharides. And so this is expressed on the cell surface. It's important for bacterial infections. It was the first toll-like receptor that we knew about and that described the function in humans. So don't worry, there are not that many of these. These are the other ones that we know about in humans. And you have toll-like receptor 5 is recognizing flagellin. 6, that we already talked about, is complex with TLR2, but I just included it here so you have all the numbers. You have 7 and 8. And 7 and 8, as far as we know, don't have any difference in function. They're two different genes, they form two different homodimers, they recognize the same thing, and they have the same outcome. Some people will say that 8 is a pseudogene, it doesn't really work, but clearly there's examples where you have both. Toll-like receptor 9 recognizes CPG motifs in double-stranded DNA. So how do you keep these all straight? Well, really the easiest way is to remember the nucleic acid ones, 3, 7, 8, 9, and 13. Okay, if you can remember that and what they do, then the other ones are a little easier. Okay, so you have 3 is double-stranded RNA, 7 and 8 are single-stranded RNA, 9 is double-stranded DNA, and then 13 is ribosomal RNA. All of those, 3, 7, 8, 9, and 13, are going to be in endosomes. Everything else is on the cell surface and is going to recognize bacteria or fungal things or some parasitic infections. And in particular, you have 1 and 2 and 6 and 2 and TLR4 are all lipopeptides, a version of those. Now the one exception to all of these is TLR10. And right now we don't know what it binds. And part of that is because this is the one toll-like receptor that probably recognizes self and provides an anti-inflammatory signal. Okay? But fortunately for you, you don't have TLR10. Oh, sorry, you have TLR10, but there's no mouse models that have TLR10. Now, fruit flies have 9 of these. Sea urchins have hundreds of these. So it's not something you can just tell by sort of the evolutionary hierarchy of how many they're going to have. It depends more on how much they use this for recognition of self versus non-self, how much they use for development versus other functions. But in humans, in mammals, toll-like receptors are primarily involved in immune recognition of foreign things. Okay, so this is one of those points that I would suggest this is a flashcard study kind of thing. Just tell me what they recognize, because I certainly will ask you on a test, what does TLR9 recognize? The questions about toll-like receptors.
Yeah, are there particular white blood cells that have the toll-like receptors? Or are they mostly on macrophages and mast cells, for instance?
They're on granulocytes, they're on mast cells, they're on dendritic cells, they are on macrophages. They also, whenever there's inflammation, many different cells, including epithelial cells, can start to express these. These are mostly innate immune cells initially express them, but then during inflammation, everybody gets a chance to recognize infection.
Quick question. In the part where it says the humans, they have 10 TLR, so they don't have 11, 12, and 13.
Correct.
Okay, okay.
Yeah. But mice have 12 different toll-like receptors, but they don't have TLR10, which is sort of what's made it very hard to study what that does. And I should also point out that TLR4 in humans is recognizing the same general structure as TLR4 in mice, but they do have some slight differences in the breadth of what they recognize. But in general, TLR9 in a mouse recognizes the same thing that TLR9 in humans recognizes. Okay, so in these other species, toll-like receptors are involved in many different processes and little or nothing to do with innate immunity. But as I said, in mammals, it's really, it really is only involved in innate recognition. Now what happens is that if you sort of look at the species, a sea urchin has a rudimentary adaptive response, but it really relies on the innate system more. And in that case, you need more toll-like receptors to provide these recognition and innate defenses. But they also use it to sort of communicate like, hey, this is my territory, not your territory, back off. So what happens? Well, Ruslan Mechitov, who was a postdoc with Charlie Janeway, who should be a familiar name if you've been reading your textbook, were at Yale, and they expressed a constitutively active human homolog of toll, and this was the first human toll-like receptor, which we now know was TLR4. And what they observed is that if they expressed this in cells, those cells would start making inflammatory cytokines, okay, in humans. Now, I should point out that Ruslan, who's also at Yale now and is a full professor, one of his former postdocs is going to be joining our faculty group in the spring, and she was trained in how in CD4 recognition of allergens. Now, I want to take a side and point out one of the people think that scientists are all just chummy and try to help each other, but Janeway died, I believe it was 2006. He should have shared the Nobel Prize in 2011, but you can't get the Nobel Prize posthumously. And so many people said, well, Ruslan should share the Nobel Prize because he was the guy in Janeway's lab that did this. They didn't give it to him. And that set off quite a feud between him and Bruce Beutler, who did get the Nobel Prize in 2011. I think for the most part, it's resolved now, but it came down to they were maliciously editing each other's Wikipedia pages and accusing each other of things at scientific meetings. Okay, so back to the science. The Ruslan expressed toll-like receptor, this toll-like receptor 4 in cells. And what he found is that those cells started making inflammatory cytokines, including IL-6 and IL-1 beta, also this protein IL-8. And this was sort of the aha moment that the toll-like receptors are recognizing foreign things, and they are providing a signal to those cells that, hey, this is how the innate system is telling the adaptive system you should do something, because it's making inflammatory cytokines. So what happens if you get toll-like receptor signaling? And so the first thing is that they stop penocytosis or phagocytosis. So if you think about this, it's easiest to think of this in the terms of a dendritic cell. Dendritic cell recognizes these, it stops taking stuff in and says, I probably have already taken this in, I'm gonna go to the lymph node and present whatever I have to the T cells, and hopefully we'll make a response to it. What does this do? Well, this says early during an infection, this is just something got in. What it prevents is, what if it didn't stop this? And so you start getting tissue damage and those dendritic cells are taking in self stuff that they're going to present to T cells. That would be bad. And so this helps ensure that it's only going to make a response to that infection. Now, for macrophages, it's just the opposite. Macrophages are not the early antigen presenting cell. So this should say, stop penocytosis for dendritic cells and start phagocytosis for macrophages. The other thing that the dendritic cells in particular will do is increase expression of the MHC proteins and costimulatory proteins so that when they get to a lymph node and they encounter a T cell, they have lots of stuff presented on their surface and are ready to stimulate a T cell. And then it'll also tell these dendritic cells that they need to move to the lymph nodes. And it does this by changing their chemokine receptors and which direct them where to go. We don't really cover that in this class. That's more detail than I think is necessary for an introductory immunology class. The last thing, this is true for both dendritic cells and macrophages, is that they'll start producing cytokines, or the proteins that are involved in activation of other innate cells, or innate and adaptive cells. Okay, so if we go back to our skin infection, just in the skin you have Langerhans cells, there's also dermal dendritic cells, and this infection then is sensed by the toll-like receptors, and these cells make inflammatory cytokines that we saw in the Medvedev experiments, including IL-1 beta. That provides the inflammatory signals, so everything else is saying, yes, there's probably something here, let's be inflammatory. And then those dendritic cells, in this case, Langerhans cells, go to the lymph node to try and initiate an immune response. Excuse me. Now, the curious thing is that virtually all of the toll-like receptors signal through the same pathway. So most toll-like receptors signal through a protein called MyD88. There's only two exceptions. TLR3 exclusively signals through a different protein called TRIF. It's toll-like receptor interferon protein. And that is a very virus-specific signal. TLR4 can signal through either of these. And they have different outcomes of this. And the main difference is what kind of cytokines do you want to make? And so even though they're all signaling through a similar pathway, it really is, what is that MyD88 associating with inside the cell that gives it a different outcome? So if you think about it, TRIF is going down the interferon response pathway. The MyD88 is going down the NF-kappa B pathway. Well, those are ultimately going to result in the production of different types of inflammatory cytokines. This again is, we recognize something, it's probably a bacteria, so we need cytokines that tell the adaptive system, make an antibacterial response. If we have recognized nucleic acids, that's probably a virus, so we want to make antiviral proteins. Now the exception of TLR4 is interesting because if you have lipopolysaccharide, that's a pretty sure indication that you've got a very serious response, and it throws the entire kitchen sink at everything, make everything. Now this is controlled by what kind of cells is expressing this and what other proteins that cell is expressing that modulates the response. But in general, all that I really care about is that you know that TLR3 and 4 can signal by TRIF, and the type of signaling depends or results in different kinds of outcome. Okay, so what happens? Well, typically, if you lack one toll-like receptor, you're not really susceptible to anything. You know, C3H mice were a little different because you're experimentally inoculating with a lot of bacteria, but other than that, they're fine. So and that's because there's redundancy. Okay, even if you can't recognize diacyl lipoproteins with TLR6 and 2, you can recognize triacyl lipoproteins with TLR1 and 2. The problem became more apparent when we looked at mice that are deficient in the downstream signaling. Because most of the toll-like receptors use MyD88, mice that are deficient in that are highly susceptible to bacterial infections. Mice that are deficient in TRIF are less vulnerable, but they are susceptible to some families of viruses. And so it's the downstream signaling pathways that are more important. Okay, so what do you get here? Well, in all cases, you want inflammatory responses. You need tumor necrosis factor. This is a general inflammatory signal. It causes vascular dilation and sort of leakiness of your blood cells so that cells can get into the sites of infection. For virtually all of the MyD88 pathways, you're getting interleukin 12. This again is a general immune response, but you need one of two proteins to generate adaptive immune response. For bacterial infections, you typically need IL-12. And for viral infections, you need a family of proteins called the type 1 interferons. And so you need one of those two to initiate an adaptive response. Type 1 interferons will also make surrounding cells resistant to infection. And then you get some other cytokines from toll-like receptor signaling, including IL-6 that raises your body temperature. So a typical question on an exam would be, name a toll-like receptor that triggers production of IL-12. Anybody give me an example?
Would toll-like receptor 4 do that?
Absolutely. Toll-like receptor 4 recognizes lipopolysaccharides on bacteria and some fungi. And so it's going to give you that signal to initiate an extracellular immune response. But it could also be TLR6, TLR1 and 2, TLR6 and 2, TLR5, any of the ones that are recognizing extracellular things. What would be a toll-like receptor that triggers production of type 1 interferon?
TLR7?
Yeah, 3, 7, 8, 9, and 13, right? Those will all give you type 1 interferons. Okay, so we'll start this. I'm not sure how far we'll get into the Rig-like receptors and Nod-like receptors, but basically they look very much the same, right? Your extracellular toll-like receptors are recognizing extracellular bacteria and fungi. Your intracellular ones are recognizing nucleic acids from stuff that's being brought in and degraded. Well, what if the virus gets into the cytoplasm and it bypasses this and gets into the cytoplasm? In that case, you have cytoplasmic sensors. And their two families are the Rig-I-like receptors and the Nod-like receptors. And these are basically there to recognize stuff that's already got in. Now for Nod-like receptors, it gets a little bit blurry because they can recognize extracellular things as well. And we'll talk about them separately. So Rig-like receptors were discovered quite a long time ago, but we really didn't know what they did. They looked like toll-like receptors in sequence level, but what they found, our Shizuo Akira found that the Rig1 protein was able to bind double-stranded RNA, and from all of the Rig-like receptors, you get a type 1 interferon response. So we don't know that they are mainly signaling through the ERF pathways. Michael Gale, who's now at the University of Washington, showed that if you put Rig-I or Rig1 at the time into cells, made them resistant to viral infection. And so that was sort of tying these two things together, that you could recognize this, get type 1 interferons, and then that would then make cells resistant to the virus reproducing. Now, the structure of these may not initially look like it, but they're basically just the same shape. They're crescent shaped, they're dimers, and they bind to nucleic acids. All of the Rig-like receptors that we know about bind to intracellular nucleic acids from pathogens that get into the cytoplasm. Now, as I said before, there's a subfamily that recognizes DNA in the cytoplasm, and there's a family that recognizes different forms of RNA in the cytoplasm. Now, you have DNA and RNA, but what they're recognizing are abnormal structures of it, or in the case particularly of DNA, your DNA shouldn't be in the cytoplasm. It should be in the nucleus. And so you have, if it's in the cytoplasm, then, and you're expressing these Rig-like receptors, then it knows there's an infection there. Right now, we know of 8 Rig-like receptors. There's not that many different forms of nucleic acids, right? Double-stranded, single-stranded, DNA, RNA, some of them recognizing caps, viral, that viruses put on them, but there are really not that many different forms of nucleic acids. Now these all work through the same pathway, okay? They all signal through a protein called IPS1. Now this is very similar to how the toll-like receptors work. They all signal through MIT88, except for our exceptions that we said. All of the Rig-like receptors that we know about signal through IPS1, and the main outcome of that is type 1 interferons, okay? They also will there's some crosstalk here that we'll sort of go over, but interferons then are released from that cell, and it makes all the surrounding cells non-permissive for virus growth. And type 1 interferons are very important for initiating adaptive response. So they tell dendritic cells, there's something here. And this is sort of an amplifying effect of saying, yes, there's something here that you need to make an immune response to. And so that causes dendritic cells to get activated and go to the lymph nodes. So what would you expect the phenotype of somebody who lacks RIG-I, for example? Well, and since we're running out of time, I'll answer this question. They're going to be susceptible to a very narrow range of viral infections. In this case, people who are susceptible to, people who are deficient in RIG-I are susceptible to mainly herpes virus infection. And that's because herpes virus makes a double-stranded RNA intermediate during its transcription. It doesn't make them susceptible to all viral infections. It's a very narrow range of viruses that express that particular nucleic acid. Okay, so the last thing that we'll cover today is how does interferon work, okay? And there's two steps to this, actually three. The first is that a cell that has that intracellular recognition of nucleic acids leads to signaling through IPS1 and transcription of interferon 1 that's secreted by the cell. It doesn't save that cell. That cell's already infected. What happens is the interferon that's made binds to the type 1 interferon receptor on the next door cells, and that increases expression of two proteins, protein kinase R and a protein called 2' 5' oligodenylate synthetase, or OAS. It's usually just called OAS. In that neighbor cell, that doesn't do anything, right? These are just cytoplasmic proteins. But if the neighbor cell gets infected, then the double-stranded RNA that's made in that neighbor cell activates PKR, and that then phosphorylates and inactivates EIF2 alpha, so you shut down translation, okay? It also, that double-stranded RNA will bind to OAS, and this makes a circular ATP that binds and activates RNase L, and you're basically destroying all of the mRNA and you're shutting down transcription. So the virus can't use anything now in that neighbor cell that has been told, hey, there's something that's probably going to infect us here. And instead what it does is it undergoes apoptosis and just sacrifices that cell so that it dies off. Okay, I think that's where we're going to pick up on Tuesday. There's just a couple more slides. We'll re-go over this interferon pathway because it is important. It is something that I will put on the test, and I expect you to know. And with that, I'll take any questions.
I guess, how do these receptors impact gene therapy related studies? If you're introducing, some form of DNA into the cytoplasm or it's consumed through an endosome, how does that, do they see an uptick in inflammation that they're not necessarily looking for because you're trying to express maybe a protein that someone's deficient of or something?
Well, in most cases, gene therapy is attempting to get a gene integrated into your DNA in the nucleus. And there are cases where you're trying to express it from a plasmid in the cytoplasm, but those tend to both work okay. If you think about how these are recognizing something, they're actually recognizing linearized versions of the nucleic acid. So as long as it's a plasmid, if it's a circle, it's actually a little tough to recognize. It doesn't bind well to any of the RIG-like receptors or toll-like receptors. However, having said that, it can eventually activate them. If that gene instead getting put into a cell, it's being cut, and then you're trying to integrate it into your host genome, then it's just a part of your DNA. It's regulated the same way, and it doesn't activate these innate receptors.
What about RNAi?
So it depends on how you're expressing RNAi, but there are examples where RNAis can also activate these pathways. There have been a number of papers where they observed an effect that they thought was due to reduction in that protein due to the inhibitory RNA. But instead what happened is they were setting off the the Rig-like receptors, and instead what they were doing was shutting the cell down by this interferon pathway. So it's something you have to consider, particularly in the lab when you're trying to do this in cells.
Thanks.
Dr. Blattman, can you go over the upcoming exam for us, or is it still coming up on next Thursday, and what we need to have ready and prepared, and do we need our cameras?
I'm meeting with Evelyn right after this, and we're gonna be finalizing those plans, and I'll send out an email today, or maybe Evelyn sending out an email.
Okay, thank you.
Professor, I had a question. What's the best way to prepare for your exams?
So this is a question I often get, is how do I prepare? And I think the worst way to prepare is to just print out the slides and look at them. In fact, I really don't like using PowerPoint. I don't do it in other classes that I teach, is because it's not active learning. So the two ways that I would study are looking at each slide and saying, okay, what are the important bits, and writing those down separately on your own. Okay, so if, for example, if we were to look at this slide here, we're currently on, what is the pathway? And it doesn't have to be extensive. You can say, here's a cell that's infected, here's a neighbor cell. What are the 3 steps? There's the infected cell senses the infection, and it makes type 1 interferons. The next door cell binds those type 1 interferons, and it upregulates these proteins. And then the third step is if that neighbor cell gets infected, then it shuts down transcription and translation. That's really, my exams are not, I don't do trick questions. I realize that there's a lot of material, so immunology is hard enough that I don't think that you need to, that I don't need to make it more tricky. The other thing that I would do is say, what question would he ask me? You know, the format of the exam is all going to be multiple choice, true and false, or matching. So what would I ask? Well, does double-stranded RNA activate PKR, and then what happens? So I'm just going through the chat. Question is, how long do memory T cells live? They're a self-renewing population. So they, on average, a T cell will divide once every couple months, but those daughter cells are with you for the rest of your life. So for viruses, it is nucleic acid recognition. If you notice, none of these innate sensing pathways are going to recognize amino acids. That's because you can vary the amino acid sequence easily and just skip out on recognition.
Professor, will there be a bonus question from this class to answer?
Yeah, but I can't give it to you until I finish the lecture, so we'll get there. It won't be today, though. It'll be on Tuesday.
Sounds good. Thank you.
So the test, yes, is still next Thursday. At 9 to 10:15. Many people ask, do we need to know who did what? Yes. Do you need to know the dates? No. It's sometimes easier to organize them chronologically, but I don't care that you know the exact date. But I will ask you who did what. And that's a big, I think a lot of science majors don't like that, but remember, there are other majors in this class that's perhaps the more interesting part. Even for many scientists, it's interesting to learn how we got to where we are.
Sorry, just to clarify, you said we don't need to know exact dates. We just need to know what they did.
Correct.
Okay.
But for me, it's easier to put them in order. You don't need to know the date, but Kangxi came first, and then Lady Montague observed that, then Cotton Mather observed that, and so they're in order, but I don't care that you know the date. Okay, any other questions? All right, we'll see you on Tuesday.
Thank you.